Hello everyone. So today we will talk about transformation. Transformation. Last time we discussed about transduction. There are three bacterial genetics, three transfers or three types of phenomena are there, namely conjugation, transduction, and transformation. Transformation is the last one, so we will discuss it today. So let's start. And transformation, simply as the name suggests, right, to transform something from somewhere to somewhere, right. In this, the we transfer the genetic material of a foreign from one from some bacterial to another bacterial cell, yeah, and the DNA that we input into the recipient, or the one which accepts that, is known as recipient, and the one that donates is known as donor, right. And how do we do it, right? How the foreign cell taken, right? And this foreign DNA is present in extracellular environment, right? It is present in the environment only, not like conjugation where physical contact was necessary and a bridge-like structure was formed known as sex pili. In transformation, what we do is that from the recipient cell, first we extract the DNA, then we purify it, and we cut down them into fragments, right? Cut down that genetic material into fragments, DNA fragments, and then it is then input into the recipient cell, right? This is the recipient cell and this is the donor cell, right? To know the why do we do this, right? To know the obviously to know the gene map of the recipient cell, right? Recipient bacterial cell, that is how the genes are arranged in that chromosome, right? As the recipient either takes up the foreign DNA fragment or use the plasma membrane of this recipient is induced or made permeable so that it can easily uptake the foreign DNA, right? The recipient either takes that yeah, it means that either the recipient takes the foreign cell on itself, right, on itself, or it is made to take that foreign DNA. We induce it, or we made that membrane of the recipient cell permeable for that foreign DNA, right, so that it can easily uptake it. Now the recipient cell is treated with an electric field, and this process is known as electroporation. What is this process called as? Electroporation. And then we'll discuss more about how this process is done. So yeah. Yes, we were talking, right, about either the cell itself takes the foreign DNA, or we induce them, or we make them by making that plasma membrane permeable. And how we make them permeable? We treat it with an electric field so that its pores are formed on the plasma membrane of the recipient cell, and this process was called as electroporation, right? Electroporation. Right? This process was known as electroporation. And now, due to which the foreign DNA easily pass through such cells, such recipient cells which are induced with electric field and form pores to take up the foreign DNA are known as competent cells. Competence means obviously it is not competent, it can easily take that particular foreign DNA. It is made competent. Now, this whole process of making a competent cell is very rare, almost 1 to 2000 to 1000 ratio, right? If there are 1000 cells, recipient cells, only among them only one will be able to induce, right? And one will that induced cell will show competence. So this is very rare, right? In ratio, remember this ratio. Next, we will talk about competence. This phenomenon is known as competence when particular shows and that competence, that it is a competent cell. Now, foreign DNA from the extracellular environment. What does it mean from extracellular environment? The first point was this only, right? It means there was no physical contact, right? It is not conjugation, right? In conjugation there was physical contact, but it is, we are not talking about conjugation, we are talking about transformation. In transformation there was no physical contact. The foreign DNA was already present in the extracellular environment already, right? It was already present in that fragmented condition, right? We extracted that foreign DNA from the donor cell, yet in the fragmented, and then it was cut down into fragments, then it was present in the extracellular environment. Then we made that recipient cell induce that recipient cell, or made competent so that it can take up. Now, transformation is taking up the DNA fragment. This is a proper definition. You can say that transformation is the process of taking up the DNA fragment by a recipient cell extracellularly or environmentally, right? Suppose this is the fragmented DNA from donor, right? Obviously, the fragmented DNA is in double stranded form. But what we do is, when we purify it and extract it, purify it during this, it is not a double stranded form. After which it comes in single strand. There are two strands you can see, A plus and A plus. What does positive means? Positive here plus means that it is a wild type. Wild type means that it can express itself, right? It can express itself, and negative means it is mutated and it cannot express itself. Here, the recipient cell, we can see recipient cell as A minus or A nothing, right? Nothing or A minus also can write. We can also write A minus also. Right. So what happens, right, when the from the donor cell the fragment comes, during which what happens is that one fragment gets degraded and only one fragment enters into the recipient cell, right? Only one fragment entered and one degraded. Here we can see only one fragment has entered, one degraded, two fragments are there, and recipient cell is already has a mutated A fragment of the genetic material. Next, here we can see now the foreign DNA or the donor DNA and the recipient DNA can pair between each other, right? It can pair between each other. That suppose there is some similarity between that foreign DNA that the donor DNA and the recipient DNA. That there is some similarity. It is not necessary there will be similarity. It by chance suppose that there is some similarity. That is they are homologous, right? They are similar to each other. Then what happens is that this homologous pair will form pair. This pairing strands, right? They will form pairs, and what will happen? Recombination will happen. Recombination takes place between homologous pairs only, right? Obviously. Then what happens is generally the pairing of four strands happen, but here we can see only three are present, right? As we know that one got degraded, right? And here only three are present, two of the recipient and one of the donor, and this recombination is taking place. What happens is this part of the and this part of that the donor stays here only, but the central part here, this part get transferred to here, this part get transferred to here, and what happens is this middle part of this get transferred to here. So you can see here, and easily see, I can make some, suppose this was totally blue. Let's do it on another page only. Suppose I can explain it here, right? Suppose it will be too light, I think. That's it. Right, A and A, and here A plus, right? So what happens is recombination takes place between them. What happens is recombination takes place between this two or this three, I can say. So see what will happen. And only the outer part had been changed. The inner part it is still A only, right? But this particular part has been changed. I'll tell you how it changed. And here it was, right? What will happen that this part of that A plus will get transferred to this here, Are you getting what I'm trying to say, right? This will become A plus, and that this part of that recipient genetic material will get transferred to here, and it will be somehow like now. This was in the recipient side, right? What will happen now? Replication and duplication will take place in that particular cell, right? So it will, we'll see it afterwards what will happen to it, right? So what will happen now, that will, it will replicate and regenerate or duplicate, whatever you like. That is quite good. So two types of form, and what will happen is one will have only the outer part of the recipient cell, and one will have only the inner part. The one which will have inner part will still be having only a and the one which had so this is yellow color, suppose this is yellow color, and the one that had that outer part of the recipient cell, it will replicate to become like this. What color it was? So it will now become a plus a plus, right? So this was transformation. In the next section we will talk about many different other processes. Thank you.